Welcome to this lecture on freshwater ecosystems. Today, we are going to dive deep into two fundamental physical properties that dictate how life thrives underwater. We will explore how sunlight fades as it travels through water and how the three-dimensional shape of a lake basin controls its biological productivity. Let us get started. To begin, imagine opening your eyes underwater in a lake. You will notice that the bright sunlight quickly fades into a murky twilight as you swim deeper. Why does this happen? Water is not perfectly transparent. It acts like a giant, thick pair of sunglasses. As light travels downward, it undergoes two main processes: absorption and scattering. Water molecules and algae absorb light, converting it into heat or chemical energy. Meanwhile, suspended particles like silt scatter the light rays, bouncing them in all directions. Together, these processes cause an exponential decrease in light intensity, a phenomenon we call light attenuation. Now that we understand what light attenuation is, let us look at why it matters so much. Sunlight is the absolute primary energy source that fuels the entire freshwater food web. Without it, aquatic plants and phytoplankton cannot perform photosynthesis, meaning no oxygen or food for fish. The depth to which sufficient light penetrates defines what we call the photic zone. This is the vibrant, life-filled upper layer of the lake. If a lake becomes too murky due to pollution, this critical photic zone shrinks. A shrinking photic zone can suffocate the ecosystem and lead to massive die-offs. This brings us to an important mathematical relationship. To calculate exactly how much light reaches a specific depth, limnologists use the Beer-Lambert law. Conceptually, this law models how light fades exponentially based on the water's clarity. In this formula, Iz represents the remaining light intensity at your target depth. I0 is the initial light intensity, right at the surface. The constant E is the base of the natural logarithm, which models natural decay. The variable K is the light extinction coefficient. A higher K means murkier water, causing light to drop off much faster. Finally, Z is the depth in meters. If K or Z increases, the light available underwater decreases rapidly. Before we move on, let us see this formula in action with a concrete example. Imagine a clear lake where the surface light is 1,000 units. The water has an extinction coefficient of 0.5 per meter. We want to find the light remaining at a depth of 2 meters. We start by substituting our known values into the Beer-Lambert equation. Multiplying the coefficient, 0.5, by the depth, 2, gives us an exponent of -1. Evaluating e to the power of -1 gives approximately 0.3678. Multiplying this by our initial 1,000 units gives a final answer of 367.8 units. Physically, this means only about 37% of surface sunlight reaches 2 meters down. Now, let us tackle a different scenario using the same law. In aquatic biology, the bottom of the photic zone is exactly where light falls to 1% of the surface light. Suppose our lake has an extinction coefficient of 0.8. We need to find the exact depth of this zone. We set the depth light to 0.01 times the surface light. Because surface light appears on both sides, we can cancel it out entirely. To solve for the depth in the exponent, we take the natural logarithm of both sides. The natural log of 0.01 is -4.605. Dividing this by our negative extinction coefficient gives a final depth of 5.76 meters. This tells lake managers that biological productivity is entirely restricted to the top 5.76 meters. Let us visualize what we just calculated. Here you are looking at an exponential light attenuation graph. Notice that the horizontal axis at the top represents light intensity as a percentage, while the vertical axis represents depth in meters, increasing as you go down. The solid curve shows the light intensity dropping rapidly right near the surface. See the dashed horizontal line? That marks the 1% light level, which is the absolute boundary of the photic zone. Everything above this line is the sunlit photic zone, where plants can grow. Everything below it is the dark aphotic zone. This diagram perfectly captures how quickly a freshwater ecosystem loses its primary energy source. Moving on from light, let us consider the physical container holding the water. When you look at a lake from the shore, you only see a flat, two-dimensional surface. However, a lake is actually a complex, three-dimensional basin carved into the earth, much like an irregularly shaped bowl. To truly understand how a lake behaves biologically and chemically, surface area is not enough. We must study its lake morphometry, which is the scientific study of a lake's physical dimensions and shape. This three-dimensional structure dictates everything, from how the water heats up in the summer to how much oxygen is available for fish during the winter. Within lake morphometry, one measurement stands above the rest: the mean depth. Mean depth is a calculated value, representing what the depth would be if you bulldozed the lake bottom until it was perfectly flat. It is arguably the single most important physical characteristic for predicting the biological productivity of a freshwater ecosystem. Lakes with a very shallow mean depth tend to be highly productive. They grow lots of algae and fish because nutrients are easily recycled from the bottom back into the sunlit waters. Conversely, lakes with a deep mean depth are usually clear and less productive because nutrients sink into the dark, deep waters where plants cannot use them. So, how do we find this crucial number? We use a very straightforward mathematical relationship. We treat the lake as a geometric solid, where the volume equals the surface area multiplied by an average depth. By rearranging this, we get our formula. In this equation, z bar represents the mean depth in meters. The variable v stands for the total volume of the lake water in cubic meters. Finally, a represents the total surface area in square meters. If a lake has a massive volume but a small surface area, the mean depth will be large. This simple formula allows us to convert complex three-dimensional bathymetry into a single, highly useful number. Let us apply this formula to a small, circular freshwater lake in a temperate forest. Environmental scientists have surveyed the lake and found a surface area of 50,000 square meters and a total volume of 250,000 cubic meters. We need to calculate the mean depth. We take our standard morphometric formula and substitute the volume into the numerator and the area into the denominator. By canceling out the 4 zeros on top and bottom, we are left with a simple division of 25 by 5. The final answer is exactly 5 meters. Because 5 meters is relatively shallow, this tells us the lake is likely highly productive and will support a robust population of fish. Now imagine a different scenario, where city managers need to know the water capacity of a massive drinking reservoir. We know the surface area is 2 million square meters, and historical records show a mean depth of 15 meters. This time, we are working the formula in reverse. We algebraically rearrange our equation by multiplying both sides by the surface area. giving us volume equals mean depth times area. We substitute 15 for the depth and 2 million for the area. Multiplying these together gives a final answer of 30 million cubic meters. Practically, this massive volume tells aquatic biologists that the reservoir has a high thermal mass and will heat up very slowly during the summer. Let us look at a visual representation of mean depth. In this cross-sectional diagram, the top horizontal line represents the flat surface area of the lake. The irregular, curved blue shape below it shows the actual natural basin holding the total volume of water. Now, notice the horizontal dashed line cutting across the middle. That line represents the mean depth. It shows exactly where the lake bottom would sit if you took all that water and poured it into a perfectly flat-bottomed cylinder with the same surface area. This diagram beautifully illustrates how mean depth simplifies a complex natural shape into a standardized, easy-to-use metric. We have covered a lot of ground today, so let us review our key takeaways. First, we learned that light attenuation is the exponential fading of sunlight underwater, driven by absorption and scattering. We used the Beer-Lambert law to calculate this fading, which allows us to define the photic zone, where all aquatic photosynthesis happens. Second, we explored lake morphometry, realizing that a lake is a complex three-dimensional basin. We saw how mean depth serves as a powerful metric, linking surface area to total volume. Ultimately, these physical properties, light and shape, are the fundamental blueprints that dictate the biological productivity of any freshwater ecosystem. Thank you for joining this lecture.